Hello everyone. So we are going to start a new topic. The title of this topic it is the structure of the DNA and the RNA and their function. So I have divided this topic into two parts, part 1 and part 2. So in the part 1 we are going to deal with the structure of the DNA and their function. So before going into the depth of this huge topic, we should know first of all regarding the DNA. So as we all know that this DNA they contain the genetic information and which is actually passed from the generation to generation. Now this DNA, as in our previous slide we have already discussed regarding the packing of the DNA, how it is tightly packed. So we can assume that what is the or how much amount of information that is being carried out by DNA. So regarding all this thing we are going to discuss here. So let's start with the structure of the DNA. So first of all, you see, if you see the background, the DNA is nothing but it is the deoxyribonucleic acid, right? Moreover, we know that DNA it is a polymer. It is a polymer of actually deoxyribonucleotides. As these ribonucleotides, they are going to keep on adding or they are going to keep on joining. They are finally going to form the DNA, right? Now, usually this DNA it is, we all know that it is a double or it have a double-stranded structure. Apart from that, they have a double helix structure also, right? The same thing only I will go in brief in the coming slide. Moreover, it is found, this DNA it is found in the chromosome, regarding which we have already studied in our previous slide. Apart from the chromosome, it is also found in the mitochondria, around 16 kb we have already discussed in our previous slide, right? And even you can find it in the chloroplast in the plant cell, right? Now it acts as a genetic material. This DNA it acts as a genetic material in most of the organism, apart from few viruses where the RNA it acts as a genetic material. And as it is a genetic material, thus we can say that it carries the genetic information, right? So how the structure has come, who have studied? So let's see some history how it has come. So let's start with a few key events which actually led to the discovery of the structure of the DNA. So first, we cannot miss the contribution made by Friedrich. So he is Mr. Friedrich. So what he has done, so actually DNA it is a acidic substance which is present in the nucleus. This concept has been put forward by Friedrich Miescher in the year 1868. But during that time, he has termed the DNA with a term which is known as the nuclein. So this is the first milestone actually in the structural elucidation of the DNA, right? The next come these two gentlemen. So they are the James Watson and Francis Crick, who are famously known for their double helix model for the structure of the DNA, right? So they have put forward the double helical model. But the thing is that once they have put forward it, they also need to have some scientific background, right? So they, apart from these two great scientists, there are few more scientists who have added or who have helped them to come to a concrete, or you can say rigid conclusion that the DNA have a double helical structure. So here you can say there are, I'm mentioning the name of the few scientists like Linus Pauling, Rosalind Franklin, Maurice Wilkins and Erwin Chargaff. So they actually helps, their postulates or their theories, they also helps to finally come to a rigid point that the DNA it have a double helical structure. So says Rosalind Franklin, and here we have the Maurice Wilkins, right? Now regarding the structure, see the DNA, the structure of the DNA it has basically divided into four types. First one it is the primary structure, next one it is your secondary, then the tertiary, and finally the quaternary, just like the proteins, right? So the DNA in its structure, it have three main components. So what are the main components? So first of all, you see it will have a pentose sugar, right? So let's start with the structure. The color is same, right? So this is the pentose sugar. Now the next component, it is the base. So there are four different ones. We know that A, T, G, C, right? So this is the base. Then we will have a phosphate group. So here you can see, or you can see here there is a phosphate group. Now regarding the nitrogenous bases, so we know that these nitrogenous bases, they are divided into two groups, right? These two groups are the pyrimidines and the purines. So first of all, pyrimidines. The pyrimidines, they are made up of one six-membered ring. It will have 6 number of carbon, right? So not only carbon, even nitrogen will be there. So the ring, it is made up of 6 member. So the pyrimidine ring, it consists of 6 member, or you can say it is made up of 6-member ring. So pyrimidine, for pyrimidine you have 2 pyrimidines. One is the thymine, another one it is the cytosine. Next, for purine, the purine, it is made up of a 6-membered ring, along with, or this 6-membered ring, it is fused with a 5-membered ring. For purine we have the adenine and the guanine, right? So first of all, you should know that the nucleotides, they are linked by a covalent bond. Known as this covalent bond, it is known as the phosphodiester bond. So what is a covalent bond? If somebody asks you, for basic understanding, I have given you here. The covalent bond is nothing but a chemical bond that involves the sharing a pair electron between atoms in a molecules. A pair electron between atoms and a molecules. So if you see the structure, this phosphodiester bond can be represented in the structure as you see here. This is a sugar, this is a phosphate molecule, this is a base, right? So this is the basic nucleotide. It is adding, or it get added to another nucleotide via this phosphodiester bond. So this is the phosphodiester bond, right? So this is the background. If somebody asks you how they are represented, you see the bases there. This is the backbone. Thymine, adenine, cytosine, guanine, right? How they are attached here. So now DNA double helix and hydrogen bonding. So this is the double helical structure, and this is the direction. So they are also known as semi or anti-parallel. So as one direction is from 5 prime to 3 prime, the other one is from 3 prime to 5 prime, right? So first of all, you should know that before going into the depth, the salient feature of the DNA structure, double helical structure. The first thing is that this DNA, it is made up of 2 polynucleotide chain. These 2 polynucleotide chain, one chain, another chain, right? Where the backbone is consists of the sugar and the phosphate. So backbone, it is made up of the sugar and the phosphate. These are the phosphate and these are the these are the sugar and these are the phosphate. Here, these are the sugar, these are the phosphate, right? And the bases are projected inside. So these are the bases, they are projected inside. So sugar, phosphate, and bases. So they are made up of the 2 identical polynucleotide chain, right? Next is that these 2 chain have anti-parallel polarity. Means we have already discussed, one will be from 5 prime to 3 prime, the other one will be from 3 prime to 5 prime, right? So now the thing is that the bases in the 2 strands are paired through hydrogen bonding, forming base pair. So we used to say like how many base pair, 100 base pair product, 20 base pair product, right? For primer we used to say it's a 20 base pair product. So what is a base pair? Base pair is nothing but a base pair. When the 2 nucleotides, they join via hydrogen bonding, they form a base pair, right? So you should know that the adenine forms 2 hydrogen bonds with the thymine, forming the opposite strand and vice versa. Thus you can say adenine, it always get conjugated with the thymine with the 2 hydrogen bonding, and for G and C there will be 3 hydrogen bonding. So I am showing you here this is the guanine and cytosine, they have 3 hydrogen bonding right here, and for adenine and thymine they have 2 hydrogen bonding. So similarly it's written here for guanine and cytosine you will have 3 hydrogen bonding. So based on the observation of the Erwin Chargaff, that for a double bond, or you can say double stranded DNA, the ratio between adenine and thymine and the ratio between the guanine and cytosine, it is always constant, it is always equal to 1. Means amount of adenine you are having will be always equal to the amount of the thymine. Similarly, amount of the guanine will be always equal to the amount of the cytosine, right? Apart from that, hydrogen bonding, I hope you know that, but for your addition, I have here again added. It is a hydrogen bonding, it is a chemical bond actually. It consists of the hydrogen atom. Hydrogen atom involved in a hydrogen bond formation between two electronegative atoms, like your oxygen or nitrogen, with one side be covalent bond and another side be the ionic bond, right? So now Erwin Chargaff experiment, what he have done. So here is Erwin Chargaff. So Chargaff actually pioneered many of the biochemical techniques for isolation and purification and even measurement of the different nucleic acid from the living cell. He have isolated many of the nucleic acid, right? So he has conducted in different experiments. So it was known that the DNA at that time, it was known that the DNA consists of the 4 different bases, A, T, G, and C, right? Now Chargaff analyzed the base composition of the DNA isolated from different species, from mammals, from birds, from different species. He have analyzed what is the composition, and what he have found actually, that was that the percentage of the adenine, it is always equal to the percentage of the thymine, and similarly the percentage of the cytosine, it is always equal to the percentage of the guanine, right? Now let's move into the structure. So you see the two strands, these are actually, they are coiled in the right-handed fashion. This is the coil structure, double helical structure, right? And this is also known as a clockwise fashion. The thing is that you see the pitch of the helix, it is 3.4 nanometer. The pitch of the helix, it is 3.4 nanometer, right? And there is roughly 10 base pair in each turn. So 10 base pair in each turn, and pitch it is 3.4 nanometer. So distance between each base pair will be 0.34 nanometer, right? That is, consequently, you can say the distance between a base pair in a helix is approximately this. You see, these are the base pairs, right? The distance between the base pair, it is 0.34 nanometer, and the distance between the pitch of the helix, it is 3.4 nanometer, right? So the plane of the base pair, they stacks over the other in the double helical structure. So you can see one, it is stacked over the other, means they are piling, or they are piled over one another, right? So in addition to this also, to hydrogen bonds, it confers the stability of the helical structure. I repeat this line because this, in addition to the hydrogen bonding, this stacking over, in addition to the hydrogen bonding, actually it increases the stability of the double helical structure, right? Now let's study some grooves. So there are actually, you see, there are two asymmetrical grooves on the outside of the helix. One, it is known as the minor groove, and one, it is a major groove. You see the difference, the gap actually. These are a groove, right? So first of all, you see there are two grooves. One, it is the major groove, another one is the minor groove. The groove, what is groove? If somebody asks you what is groove, it is nothing but it is a furrow, a slight depression. It is just like if you see a river, it is flowing through a plane of soil, you will have some groove. Once the river get dried up, right, you will have some furrow, or you can say some depression, right, or channel. So this is the groove. So basically the certain proteins, they can bind with this groove. That's why the study of the groove, it is also necessary. So they can thus interact with particular sequence of the bases. This group can interact with a particular sequence of the bases. Now you see here, 3 major forms of the DNA are available. What are they? They are the B, A, and Z form of DNA. These are the 3 forms in which the DNA exists. The first form, it is the B form. So see, it is the most common form. Most of the human body, means human being or any living organism which have a nucleus or which have a genetic material, they generally will have not only nucleus, because nucleus is non-nucleated in human cases, right? So basically all the cells which have a genetic information, means a nucleus with the DNA or RNA, or you can say simply chromosome, they will have the B DNA. It is the most common form of DNA, means DNA. Now regarding the B structure, see, it is actually It is a helix, meanings that it has a right-handed or clockwise, or you can say spiral structure. A helix, or you can say alpha helix, right? Moreover, complementary, it will have complementary base pairing, right? Apart from that, similarly only A will bind to the T, G will bind to the C, right? Now the ideal bDNA has 10 base pair per turn. So whatever we have studied now, just now, it will apply directly to the bDNA. And per turn means you will have a 360. This is not 3600, it is 360. This is the sign for the degree, 360 degree rotation of the helix, right? So each base pair is twisted 36 relative to the adjacent bases, right? The base pair are, we have already studied regarding this thing, the base pair are 0.34 nm and the distance between them will be 3.4 nm, right? So the complete rotation of the molecule, it is 3.4 nanometer. The axis, there will be axis, arbitrary axis, that axis will pass through the middle of the each base pair to divide it into two equal form. The minor groove is narrow. In B form, you should know that the minor groove is narrow and shallow, this one, whereas the major groove is wide here in the B form and it is deep. The next thing is that this structure exists when plenty of water surround the molecule. So B form of DNA you will get only when there is a plenty of water, and which is most common in a living cell. Thus you can say that there is no unusual base sequences in the DNA condition that are likely to be present in a cell. So thus you can say in a living cell or in a condition where there is a plenty of water, B form of DNA it predominates over the other forms. So the bDNA structure is most stable configuration for random sequence of the nucleotide under the physiological condition, right? Now you move to the second structure. This is the A form, that is the aDNA. So it is also a right-handed helix. It is wider and flatter as compared to that of the bDNA, right? Here you will having, earlier we are having 10 base pair per turn, right? But in the A form you will have 11 base pair per turn, right? It was means actually its bases are tilted away from the main axis of the molecule. If we suppose this is the main axis of the molecule, earlier the bases were similarly placed here, right? Now the bases, they will tilt, they will come quite apart or away from the main axis, right? Now the groove nature here actually in the A form you will find narrow, deep, major groove, narrow and deep major groove, whereas broad and shallow minor groove, right? Then A form of DNA, it is generally found in the dehydrating condition, means when the water level or amount of the water it is less in the cell. So the aDNA has been observed in two contexts, in two contexts you can find the aDNA. First, when in the active sites of the DNA polymerase you can find the aDNA. Moreover, you can find it in the gram-positive bacteria undergoing the sporulation. So you will find the if the gram-positive bacteria undergo sporulation, you will find this aDNA, the zDNA. This is the next part. So here also it is a left-handed helix. The structure, it has a left-handed helix. It is seen in the condition of the high salt concentration. When the DNA it is subjected to a high salt concentration, you will get the Z form of the DNA, right? In this form actually the sugar phosphate backbones, earlier we have studied, right? The sugar phosphate backbones, they will zigzag back and forth. The movement, their direction, they will zigzag. Earlier it was linearly arranged, right? Now they will zigzag. Linear means back and forth, giving rise to the name, the zDNA, or you can say it's a zigzag DNA, right? Here you see per turn you are going to have 12 base pair. So they will have a deep minor groove, but you can say no discernible or major groove. Major groove it is generally not observed here. So part of some of the, it is a part of this Z gene, it is a part of most of the, some of the active genes form, right? So zDNA suggesting that the zDNA may play a role in the regulating gene transcription. So there are many theories that has been put forward as the zDNA, they are a part for regulating the gene transcription in many models. The next concept you should know that or you can say the last concept for DNA, it is the DNA supercoiling. So regarding which we have already discussed. So DNA supercoiling is nothing but high amount or highly packed structure of the DNA. So DNA supercoiling refers to the over or underwinding of the strands. So you see the DNA supercoiling, it is important for DNA packaging within a cell, because we know that if we keep on unwinding the structure of the DNA, you will have kilometers and kilometers, right? So because of the length of the DNA can be of 1,000 times from that of the cells, thus packing this high amount of the material into a cell, it is difficult. That's why DNA supercoiling it takes place. So thus you can say the supercoiling of the DNA, it reduces the spaces and allows for much more DNA to be packed within the cell. So this is all from the DNA. So thank you everyone. Hope you like this video. If you like this video, just press the like icon. You can share this video with your classmates and colleagues, and for further notification from this channel, you can press the bell icon. So thank you once again.